We do have time for a short panel discussion about takeaways from our event this week, and we've got a few people who can be part of that discussion. So we met Melanie this morning. So in the interest of time, I won't reintroduce. We also have Pangkong Fox. Pangkong is science director of the CACNA 1A Foundation. Leveraging her background as a scientist, educator, and rare disease mom, Pangkong facilitates the integration of patient-reported outcomes into the foundation's strategic research roadmap and coordinates the execution of the research network and the foundation's scientific initiatives. Pangkong has a PhD in cell and molecular biology and is motivated in her work by her son Alex. We also have Dr. Barbara Bailus, who's assistant professor of genetics at the Keck Graduate Institute of the Claremont Colleges. Doctoral research was done at the University of California, Davis, in the Siegel Laboratory, developing a gene therapy based on artificial transcription factors for the treatment of Angelman syndrome. Postdoctoral research was in the Ellerby Lab at the Buck Institute for Research on Aging, focused on neurodegenerative disorders. Dr. Bailus has also been chair of the Scientific Advisory Board for the Foundation for Angelman Syndrome Therapeutics, so really understands the patient advocacy world. I apologize, Barbara, I'm going to be a little brief on your bio, just in the interest of time. Joe Farris, who has been helping with Q&A, is a genetics fellow at Mayo. So starting with Peng Kong, as you've been listening along this week, what are your impressions and takeaways that you think we as the UGDH community should be thinking about?
Thanks, Rebecca, and thanks for inviting me to take part in this. I really have to say I'm so impressed. I met Rebecca about 3 years ago when I think everything was just starting, and I remember you saying, "What do we, what should we do?" And here you are, 3 years later, already working and thinking through a gene therapy. You have wonderful preclinical assets, which is just a fancy way of saying you've got these cellular disease models and the organoid models and mouse models. So I think that your community should be really excited and really motivated, hopefully after this, to really come out and share their stories. I think that's a big thing, especially with an ultra rare disorder. Sharing stories leads to data. That data is what drives the research, and the research is what leads to treatment. But with this ultra rare disease, for any rare disease, it all comes back down to the information that we can learn from those living with this disorder. And I think that you've really, the UDGH Foundation has really done a great job doing that. And you've run in such wonderful people. I'm, of course, biased. We work with Lena Lusk, we work with Dr. Varignano, and I was really glad to see them, joining your medical and scientific advisory board. I think there's a lot of progress being made. And I don't think I have to really, it sounds like your researchers and your clinicians see the urgency. And my biggest takeaway for the community is to just, tell your stories. Every little detail matters. And to the parents out there, you are experts on your kids. And like Dr. Barignano said, you might not have all of the medical terms, but just, tell your story. And those medical terms can be integrated at some point. But the best way to understand how to build the right treatments and how to design the best clinical trials really comes from you.
Thank you. Barbara, do you have thoughts on that question?
Yeah, so I would agree on being completely new to the UGDH community, not having heard about this disease until maybe about a year ago. I would say I'm very impressed to see the cellular models because there's very robust phenotypes in those models, which makes treatment screening extremely helpful, to have something that you can model that easily. And also for the mouse models, it's impressive to see that you are taking on the challenge that the original knockout was embryonic lethal and finding different ways to work around that. Because really, when you start going down the path to designing a treatment, the FDA likes to see two things, which is something in human cells and something in a mouse model with behaviors that you can recover. And once you have those two, and they're characterized well, you can really start moving toward finding different treatments. And I would say start moving on finding the treatments as soon as you can. it takes much longer than anyone usually imagines to find those. So it's never too early to start.
Melanie, what were your impressions and takeaways from um, this week in particular, and anything, in general from your work that maybe has you weren't able to highlight yet?
First of all, I'm very, very impressed by all of the research that I've heard about and all of the talks and all of the families that were part of this symposium. And so I really appreciate that everyone shared whatever their participation in the community is. It's really pretty awesome for someone who studies UGDH to have a community that's focused on UGDH. And I'm very excited about being part of the community for that reason. Thinking about UGH, some of the aspects of its regulation that I didn't have a chance to really talk about are the post-translational modifications to it. And so what kinase cascades crosstalk to influence the activity of UGDH and how it directs UDP glucuronate into its various downstream products. We've done interactome screening, and so we know that there are multiple interacting proteins that do govern some of the activities of UGDH, probably selectively. And so, as we're thinking about therapeutic options, it's not just going to come down to gene therapy. We need to understand what is the role of UGDH in really specific conditions of extracellular stimuli and nutrition availability, et cetera. What effect does that have on the specific downstream function of UGDH? Because if you understand the protein-protein interactions, that's an occasion to figure out how you can design selective inhibitors or selective activators where you just are channeling the product into a single fate instead of having it regulated into multiple competing fates. And that has the potential then to be individualized with respect to its treatment of patients that have different variants.
Joe, how about you? Do you have comments on takeaways from this week and where we are in this odyssey?
Yeah, for sure. I mean, I think my major takeaway, just to speak to the patient families, is that um, I think every day you're asked to do the impossible, in that you're asked to wear so many different hats of being, first of all, a caregiver to your child. You're asked to wear a hat of understanding extremely complex biochemistry and genetics that probably you thought you'd never have anything to do with. You're asked to navigate complex relationships between industry and research funding. Um, so it's been extremely inspiring to see all of you stare into the face of that impossibility of having all of those roles while also being a caregiver, and accomplish so much in such a short time. So I'm extremely encouraged. I think this group is extremely inspiring. It's been my privilege to serve on this board for a brief time and hopefully a much longer time in the future. So uh, I'm just extremely encouraged by seeing everything that's been accomplished at this juncture.